I think just in the interest of time, we could get started since we've got a lot to go over today. Want to welcome everybody to iFyber's fifth webinar in this series, I believe, and this is going to be presented by Dr. Marielle Medina-Reviera. I know we've got a lot of kind of friends and colleagues on the call that know us already, but I do want to just quickly introduce iFyber. Marielle, if you could advance the slide just one, or maybe two. So iFyber is a contract research organization with a focus on custom method development and research activities. We've been in business for 18 years, and throughout that time, consistently worked at the intersection of the disciplines that we think are most important to the medical device and pharmaceutical industries, biology, chemistry, and materials. That's where we spend our day to day working with innovative developers like you all, trying to move projects forward from point A to point B. A couple of just brief housekeeping notes. We are going to record this webinar. So if you have to drop off, there'll be a link to it later that you can come back to. Please keep yourselves muted while the presentation's going on. If you have a question or any questions, please put it in the chat or that Q&A button, and we'll handle it kind of according to how time allows, either while Marielle is going through the presentation at the end, or if we don't have time, we'll follow up with you over email. I'd like to introduce Marielle. Marielle is a senior research scientist and technical lead at iFyber. She earned her PhD in microbiology from Cornell University, has a deep experience in microbiology, molecular biology, bioengineering, and in vitro method development. Marielle joined us in 2020, initially as a postdoctoral researcher, and then she soon transitioned into a full-time research scientist position after that. So Marielle leads design and execution of mostly preclinical studies. You'll see today's focus is on laboratory studies for clinical studies, but most of the work we do is in the preclinical side, supports healthcare, biotech, and medical device clients. Really a focus on antimicrobial efficacy testing, biofilm testing, biomaterial characterization, and these clinical research applications that we'll spend time on. Her current work interests of late center on developing anaerobic-based in vitro models. It's been a big push we're not going to talk about today, but it is exciting that we've been developing that and evaluating antimicrobial materials against the clinically relevant anaerobic microorganisms and biofilms that are difficult to get to because they're anaerobes. With that, Marielle can get started. Thanks for your attention and for joining us today.
Yeah, thank you so much. So the title of this presentation is Integrated Tools to Evaluate Biomarkers and Microbiota in Clinical Studies. I would like to start with just key discussion points that we are going to go over the hour. So first, I would like to define what I mean by integrated analysis. Why does that matter? What is our approach to such projects, and just give you some examples in terms of case studies that we have done over the years, and then just summarize key takeaways. So first, how we look at the clinical studies that we work with, we don't run the studies. we work with a clinic, we work with a sponsor, we have a cross-disciplinary laboratory, meaning that people from different disciplines in biology and chemistry with background in material science that can support each other and providing a very well-rounded experimental design to answer the questions of the sponsor that is definitely connected to what's happening in the clinic. The clinic is the one who is seeing the patients, selecting them, taking measurements that are really important to understand what's going on with that patient and important for the goals of the study, but it needs to be translated or interconnected with all the assays that we can do in the lab. So for us, integration is that portion when we look at the goals of the sponsor and then design an experiment or a study with a clinic, with a sponsor, and our lab, and our resources, our analysis tools to be able to answer the question. The questions sometimes are very straightforward and there's only one analyte. So we are looking for bacteria in a biopsy of a wound. So that is very specific, very like point driven, but sometimes we want to see a little bit more than that. We want to see biomarkers. Is the wound closing? So it is a more rounded, and you might need people from other disciplines to join the laboratory team to be able to answer those questions that might be a little bit more about the mechanism of action of the treatment versus present and absent of a microorganism. And altogether, all those disciplines will give a stronger clinical evidence, again, that will support the goals of the sponsor, but also interconnect with what the clinician is seeing with the patients. So I would like to divide the next sections into things that I believe are very critical for us to manage that goal, and is my audience. And by audience, I mean a little bit the sponsor, but also the end user of that product, of that material, of that device that we are evaluating. Then it's like also the considerations related to the sample. What is the sample that we are collecting? What are the analysis that we are going to conduct? What are we looking for in that sample? So I will just go a little bit about those aspects. So first is what I call the audience and the endpoint of the results. We often have sponsors that reach out to us with a study that is designed to inform some development. Development could be improvement of a material that already exists or a device that already exists, but also something that is on the market. Sometimes it is regulatory, it's just gathering as much information as possible for a submission, but sometimes is I want to know how my product is doing compared to others. So the science still strong. The experimental design will be very similar, but the data analysis, the plots are the narrative just take you to a story that is different just because it's a different audience, and the questions and the goals might be driven by that audience. Moving on, the second portion that I mentioned, the sample, and this is really important, and it's important that the sponsor and the clinic involve us really early on, because it's not the same saying, oh, I want a biopsy, but then how big is the biopsy, or how big the biopsy needs to be for me to be able to isolate enough bacteria to produce strong data? How big is the swab, like how much of the exudate you need to swab, and where are you going to store it, in what media, at what temperature? All those things are critical for the lab group, for the people who have done this before and understand the system to be able to weigh in and provide information about those decisions really early on. We work with a variety of materials and tissues. Also, we source the proper media, the proper containers to be able to store appropriately, and for the samples to ship into the lab on time. There's going to be samples that needs to be processed within 24 hours. There's samples that we manage to preserve them for longer periods of time. So all of those considerations is just saying, I'm going to take a swab, is what swab, what materials, where you're going to put it, how you're going to store it, what are the proper labels for the boxes to get to the lab. All of those considerations get done in combination with that integrated group of people that is the sponsor, the clinic, and the group in the lab. Another thing that I wanted to mention and go over is the analytical target. What is my target? Am I looking for bacteria? Am I looking for the host response to the treatment, to the presence of the device? We have to know that to be able to understand what's coming next, which is the analytical tools of the methods, how we select them. If we are looking into the microorganisms, so then we have to think about, what am I doing? I'm going to culture, I'm going to do PCR, am I going to sequence? Then if I'm interested in the host, so then what biomarkers I'm measuring. Is it wound healing? Is it rejection? Is it inflammation? What is it that I'm looking for to be able to measure? And then tissue analysis is something that we as well do that will allow you to understand also the host response, but it's a different set of techniques. So it's a different group of people in our team that can weigh in on the proper preparation of the sample, the amount of sample, and also the analysis of the results to select the proper type of stainings that will give you more information. All of this, whatever tools you decide, or we as a group for the goals of the experiment, we decide it needs to be somehow interconnected with the clinical endpoints. So if you have an endpoint in the clinic that is associated to the wound closure, so what are the biomarkers that are measuring here are telling you at the same time at the same day of collection? And how they are combined to each other. This might be a very busy slide, but it's on purpose because we are a multidisciplinary team. So based on the goal, we can look into what is transcribed and produced by the host or even the microorganisms during that treatment. We can look at the proteins that are produced. We can look at the metabolome. We can look into the microorganisms that are there doing something at the process of the treatment. And then with that information, combine all of that and translate into things that are related to what's actually happening with the patient. So that is kind of like the background in the interaction. And I would like to move forward with case studies. And the idea of the case studies is to provide some examples of all the things that I talk about, the relationship between the goals, the audience, the sample, the analyte, and the analysis tools. So I would like to start with this comparative evaluation of 2 wound care treatments. We have similar studies. I picked one. So in this case, our sponsor, the goal was for marketing. So they already have this dressing in the market and they want to see how it compared to the other treatment on the market. So we ended up with treatment A and B. Samples were collected at 3 time points, 14, 28, and 42, that they mean something for the wound healing process. The clinician collected swabs of the wound, the exudate, and also biopsies. And that collection and all that process was done together between the clinic, the sponsor, and our team. So we prepared the right kit for them to do that collection. So once the samples arrived, so the swab were intended for bacteria load testing. Then the biopsy was intended for wound healing biomarkers. We were interested in the transcriptome. I'll expand on the next slide about that. And then also visualizing the tissue. So we did some histology for this client with some of the biopsies that were collected. And the idea was to, again, compare treatment A with B over three different time points and look for microorganisms, but also the host response. For the histology portion, the biopsies collected were used to evaluate the general tissue morphology. So depending on the treatment, so one tissue will look different than the other, and depending on the treatment, there's some changes in morphology that happen while the wound gets closed and healed. So those can be visualized with a stain, like H&E in this case. There's other stains available, but in this case, H&E was selected. We have done mesotrichome, that you have an example there on the right. We have done PAS as well. In addition to H&E, we did some immunohistochemistry, specifically for the C31 that provides some information about angiogenesis. Again, another biomarker in this case using more like a visual technique to see how the wound is progressing. For the transcriptome, so this is quite interesting, and this is one of those techniques that I love so much. So I'll go over a little bit deeper on that. So the transcriptome is a complete set of the RNA molecules that are transcribed and present in a given cell, tissue, organism at a specific time. So in this case is the wounds that receive treatment A versus B over time. And the idea for this is to understand what was happening through time. So there's several methods, tools that you can use that are out there. So we presented 4 options to this client and other clients that we received before. And it depends on the goal. So it's always a balance between the budget, the time that is available, and the goal. So if a technique that is a little bit lower in cost or it's less costly can provide enough information to answer the questions, so sometimes is what happens. And this is option number 2. So we have done PCR microarrays quite often. So the idea with this is you extract the RNA from the tissue that is provided by the biopsy. You amplify that to get a signal, and you target specific wound healing markers that are also known as biomarkers. In this case, the one that we selected, there's other panels available for us to order and work with. The one that we selected contained 84 key genes that are central to wound healing. It is supported by publications. There's a full understanding in the community that works with that type of work of what those genes indicate, if they're up or lower regulated. And just to give you an idea of what they're telling you, those biomarkers. So for example, some of them will be telling you inflammation or cytokines being produced, are growth factors activated, deactivated, how they compare between the 2 treatments. So it's like a data set that is quite comprehensive. And it's a lot to analyze when you receive the samples from various patients. So what I did here is a summary of the result, but it's like a massive summary of all the data that was captured in this study. So for histology, we learned that treatment A enhanced tissue repair. So the repair happened faster. This is based on the comparison of the images of the stain. We saw closure faster. For the biomolecular markers that match the same idea of treatment A, producing a faster closure, there was an enhancement in the adhesion, proliferation, and angiogenesis for the cells, and the extracellular matrix was started to remodel at around 4 weeks, which was faster than the other treatment. That with the microbiome analysis, so we observed a reduction of the bacterial bioburden over time. So it went down to a lower concentration for treatment A than treatment B, as shown by the plot on your right. You can tell that the starting concentration was comparable. At day 24, it was about the same. And then the last time point is where we saw like a differentiation between the two groups. Like many of the clinical trials that we support, we are blinded, but we don't know what treatment A or B are. So we were very happy first to learn that the results correlate with what the client was seeing at the clinic. So all the indicators that or the biomarkers that we evaluated were telling the same story as the client, but also that treatment A, which is their product, was performing better than the competitor. So it was a double happy type of end to this study. So I would like to move forward with this other one. The second one, we were evaluating, in this case, the bacteria attached to a bariatric device. The device was implanted and then explanted from the patient, and then we used that as a pilot to evaluate or develop a good method to identify the bacteria that was attaching to the in creating a biofilm or attaching to the device. There's another one that is going to be wound healing. That's the third case study. But I want to go over this one first. So as I mentioned, it was a bariatric surgery balloon or device that was implanted. We were supporting the R&D team, which had this device out of and wanted to understand how it was performing in terms of the microorganisms. They had a set of analysis tools at their disposal, but they wanted to understand the bacteria population and the fungi population that was attaching. So the idea is we prepare a kit, distribute that across the different clinics that we're working on that. And then a part of the process and part of the sponsor goals was to understand if the bacteria and the microorganisms in general were attaching more to one segment of the device than others. The image that you see on your right is an image from the internet. It's not necessarily their device. It really doesn't mean that that device look exactly the same. So there were some differences in the design and the materials from the top to the end of the device. During the process in this pilot study, we learned that the first two portions of the device did not have a lot of microbial attachment. So the results that you're going to be seeing pretty soon only include section or segments 3 to 5. The methods and the tools that were selected for this was next-gen sequencing of the 16S and the ITS. And I'll go over that here. So like other analysis tools, we with the sponsor work around what is available in terms of techniques. We presented the option of doing whole genome sequencing. It is more costly, but it provides a more comprehensive idea or results of the population, and it allows you to distinguish up to the species level. On the other side, also a next-gen sequencing technique that only goes to one gene, 16S or ITS. 16S gets used to identify bacteria, ITS it's used to identify fungi. genera. So the idea here is you pick a gene that is highly conserved, that will give you as much information as possible, and move forward with that. Because that information is enough to answer the questions that the sponsor has. This is just to kind of like go through our study workflow and what happened in this one. So we prepared that sampling kit was specific to be able to protect the devices from transport. And then they came to the lab. We developed a DNA purification method that was exclusively to isolate the microorganisms that were attached to different segments of the device, not the planktonic bacteria that was surrounding the device. Then we performed some quality control assessment that gets done all the time before sequencing. Then we move forward with next-gen sequencing of the ITS. As I mentioned before, the ITS is for fungi, the 16S is for bacteria, and then the bioinformatic analysis to make sense of the data. The next slide is start the results. It was a big study. A lot of data came out of the bioinformatic analysis. So I selected some plots that are just examples of all the information that was that we got out of that. So basically, I'll go over the heat map. So this is a heat map, very common in sequencing results. The results can be presented differently, but this is the standard. Here I have the device segments. So 5, 3, and 4 are over here. Over here are the microorganisms, in this case, fungi, that were identified in that population. The color code referred to the relative abundance. Darker means that that particular genus, and in this case up to species, was present in a given device at a higher abundance or concentration than the others. This is within that segment, and this is kind of like color coded between the segments. I'm highlighting here, over here, Candida. And Candida, we have Tropicalis and albicans. They represent most of the fungi population that was identified out of this patient, followed by Pichia with some other species that might be relevant, such as Mucor and Saccharomyces cerevisiae. Candida being dominant in this population was something that we highlighted for the sponsor. And we look at the literature for all the species that were dominating the community for fungi and for bacteria. And Candida was important because there's a lot of evidence in the literature that a microbiome in the gut that is dominated by Candida might translate into a patient with chronic bloating, gas, abdominal discomfort, diarrhea, and constipation. So that is an important information that was given to the sponsor to make decisions about that and give that to the clinic as well. The rest of the microorganisms that we isolated, the rest of the fungi that we isolated or identified, they were commensal organisms that are known to be present and being part of the microbiome, but just being opportunistic pathogens if the environment provide that stress. Similarly, we work with the 16S and got the bacterial microbiome. So again, this is a heat map. This is the color code for the relative abundance. So darker means that there's higher abundance of that particular species. Over here, we have Lactobacillus, which pretty much it's very common that Lactobacillus is known for being a positive microorganism that is part of our microbiome. And it was the most abundant genera out of all the ones that we saw. present over there. The other ones that are common members of the Bacteroidetes and the Firmicutes, which are groups that are important for the microbiome balance. What we learned from the bacteria side of things is that this bacterial microbiome was dominated by commensal gut microbiota. Nothing stood out of all the microorganisms that we saw. And all of that was communicated to the sponsor so they could make decisions about this patient and have a better understanding of what microorganisms are able to attach to the device and make decisions about the device design and future improvements to this. So moving forward, I would like to go back to wound healing. We do have a lot of sponsors coming to us for different projects. This has been in the few years kind of like a recurring experimental design where we work with point of care equipments or devices. In this case, this one produced a fluorescence, change in fluorescence that you can see. And if the wound typically have high concentrations of bacteria, so the fluorescence is higher and the clinicians will be able to make a decision. So the idea with this specific study that we completed about a year ago was to determine the bacteria bioburden. So if the point of care device that produces fluorescence is doing the job and is working as expected, so the fluorescence will be produced if the bacteria is at a concentration of 10 to the 4 CFU per gram or higher. And then the question is, if the fluorescence is telling you that, what is telling you from the biopsy that you collect at the same time point? So the clinicians collected the biopsies, but in the kits that we provided, they got sent to us. And then we evaluated the bioburden. For this particular project, we did aerobic bacteria. We selected a specialty media that will be able to differentiate between gram positives and gram negatives, and also specialty media that it was specific for anaerobics, and we use anaerobic boxes to be able to grow those. So it was a well-rounded to try to understand if there's some bias in the equipment, depending which group the bacteria belong to. In terms of results, so 51 of the samples or the biopsies that we received were positive, and 49 were negative, meaning that they had a concentration of less than 10 to the 4 CFU per grams. We were blinded again in this study, so we were very happy to learn that the data align what they were seeing over there in terms of endpoints and how the device was performing at the clinic. We completed last year another project similar to this, and there's an ongoing project similar to this that is for 100 patients right now. And so the last case study that I want to chat about or share information is this one. It's very exciting for me. It's one of those that it's just very, very interesting to know that in biology and sometimes core science, there's stuff that we still don't know. So we got approached by a sponsor. They are the manufacturer of a device. This device get in contact with this material. So there's not a lot of publications about it. There's not an artificial biomimicking fluid that they can purchase to evaluate their device or their raw materials before they become devices. So it's a big problem because you have to go to like an actual clinical trial to use your device before you can test the device with a fluid and do a little bit of playing in the lab and screen for things that might be completely outrageous and will be really bad. So it is a very, very important decision or a very important piece of information to have a well characterized matrix. Without that, you might make big decisions that impact in the long term. So this is a multi-phase study. So the first phase is basically characterize that matrix. We were supporting the R&D team, and we brought in our broad team. So we are talking physical properties, chemical properties, and biological properties. So physical, the viscosity of the material, the surface tension, what is the pH for the chemical, the liquid solid ratio, how much proteins are there, what proteins are there. And for the biological, that might come a little bit later, but understanding the redox potential, this is important for the microorganisms, what microorganisms are there, the transcriptomes from the host that is producing. And we ended up using a ton of different methods or endpoint analysis strategies or tools, colorimetric assays, LCMS, next-gen sequencing. We can use RNA microarray. We can do culturing for the bacteria, further characterization of the proteome, the transcriptome with different techniques. So this is ongoing, and it's one of those that is very exciting because this information from at least 100 patients that we complete the whole set will provide enough information for this client to either create their own artificial substitute for analysis, but also select the right materials in advance, really early on, and just make decisions on time. So this is just an example that I wanted to share about our collaboration and how we integrate kind of like what the clinic is doing and the work that we do with the clinic who collect the samples with the sponsor goals, and then our team, our broader team, making decisions over here. So just connecting that project with this slide is more that connection in turn, like you have a goal, you try to find the right techniques and the right people in the lab to be able to answer those questions. And that's where a truly multidisciplinary team comes about. So we could work with the DNA, with the RNA from the host, the proteins that are being produced, the cells, the tissue, and use many tools at our disposal to be able to understand what's going on and target the goals of the study. Our team is not just multidisciplinary, we go beyond the raw data. So we provide full reports with data analysis, we prepare posters and write peer review publications with our sponsors. We also assess on marketing materials that you sometimes see on the conferences. Also, I wanted to share a little bit of the other work that I do, just real quick, because often we are part of preclinical studies that serve as a prelude to clinical studies. We have good translational preclinical models that help our clients down select products, sometimes at the concept stage, prior to the actual clinical assessment. Clinical trials are expensive. I'm not going to lie. They are long term. So this is months and months of work from the lab, the clinic, the sponsor, sometimes multiple labs depending on the needs. So if you have really good models, you can down select your options and narrow down the experimental design so it will be more direct to the point. So just a few takeaways of the things that I already mentioned. So an effective clinical testing start with informed scientific decisions regarding the sample collection, the analyte, the analytical methods that get used. And this is just to ensure that the goals are met and they're clinically relevant and useful for the sponsor, not just measuring something for the sake of, or just selecting the right technique to do that. And for you to be able to do that, you definitely have to involve a multidisciplinary team with the right approach. The combination of the sponsor, the clinic, and the lab, but also the group of people that have the background in biology and chemistry and physics, material science, to be able to select some of these techniques, but the materials and the supplies that are going to be used. And then I just want to stress the importance of involving the lab really early on, because some of the decisions, such as how much sample to collect, is something that might need to be there at the IRB submission. So all those things that are really important to be decided really early on, it's important to ask the scientists who are going to run the experiments to make sure that you can get the data that you need, or the sponsor will get the data that they need and meet their goals. So early integration between the three teams is, I believe, critical, and it's just going to make a stronger set of data and make sure that the project is on budget throughout. I want to thank the iFyber team and our sister company, Nova Sterilis. We are a multidisciplinary team that ranges between biology, chemistry, and material science. I'm on the biology side and molecular biology. And I'm all done. So I'm happy to answer any questions that you might have for me, for my team, and just let me know.
Are there any questions from the audience? We've got a few minutes, so feel free to unmute yourself and chime in. If you're thinking of a question and you need a second, I've got one for you, Mary Ellie. Is there any way you could speak to, you mentioned the amount of sample and how you collect the sample, but the transport of the sample from the sites to the lab, I feel like that's a part of it that kind of gets maybe looked at late sometimes, the act of transporting samples. Can you speak a little to that?
Yeah, of course. So it depends a lot on the type of sample. Sometimes you need to freeze them immediately. You need to store them at 4 degrees. So it depends a lot on the type of sample. So what we do is try to figure it out. What is your goal and point testing? What type of sample are you planning to collect? And then what do you need then for that? So what type of vial, what type of media will preserve the RNA that is present in that sample, and the vessels or the containers that are more appropriate for that. On top of that, there's regulation related to transporting biological samples through mail. So we have to be in compliance with that. So that touches on secondary containment, proper labels, tertiary containment, the coolers, the boxes, the labels that go out. So we definitely, it's important for us that sponsor involve us early on so we can provide that information and assess either source all those things ourselves and provide a checklist or instructions on how to handle them, or assist them in the selection themselves and handle that themselves.
Thanks. Any other questions from folks? Okay, well, I'll take the silence to mean that everyone was satisfied and enjoyed the talk. I appreciate Marielle for putting this together and for everyone's attention today. If this is coming up for you and your organizations as you guys start looking at clinical trials, I know especially in the wound space, there's been a lot of interest in running clinical trials given the regulatory and the payment ends of things in the world over the last couple of years. We are open to chatting about it and working with your clinical CRO or your regulatory team to talk through these types of studies and see if there's any way we can help. With that, I'd like to thank you again, and we'll be concluding the webinar. Have a great day, everybody.
Thank you.